to work. All right. Well, I'll wait a little bit before I do the announcements. Let's see. All right. Well, we've got 1 minute left, so I'll go ahead and start the announcements since this is being recorded. And most of this is going to be repetitive anyway. I hate to repeat myself, but even though I'm repeating myself, some people seem to have not gotten the message, so I'll keep saying it. First of all, I'll remind everybody to email me for attendance. If it's after 9 a.m., it's late. Since I'm not requiring you to be here, that's like the way I can make sure that you're at least up and pretending to be paying attention. So yes, make sure you email me for attendance before 9 a.m. All of you, when you get a chance, go check your MyState. Everybody online. The one person in here, I know it's not an issue, but you need to check your MyState because there's one of you who is not on my roster. So one of you is attending class and has been kicked off the roster, not by me, but apparently by the university. So you might need to check into that. There might be some sort of financial aid issue or something. So yeah, all of you, please do that. What else? Hopefully today we will finish chapter 3. And once we finish chapter 3, that means we're done with the first 3 chapters, obviously, which means we will do our first exam. So the first exam will be sometime next week. So keep an eye out on Google Classroom this weekend, because if we do, whenever we finish chapter 3, I'm going to send out a message and ask you when you want to take the exam, whether it's Monday, Tuesday, or Wednesday, and I'll kind of take a poll. And if you could, when you tell me which day works best for you or which days don't work best for you, if it's a really important reason, please include that. Like if it's something like, well, you know, I know I'll be in the doctor's or I'm getting surgery Wednesday morning, so it can't be that for me. That's one thing versus somebody be like, well, you know, Fridays I usually it's hard for me to wake up. But at least tell me that. It can be honest. But anyway, respond to that when you get it. And then the usual announcement is that you always hear. Don't forget independent work. Some of you haven't done any. I really, really recommend getting it started, at least the study guides. What else? And also remember, this week's lab is due tomorrow. I pushed it back a day since I didn't give you your lab until Tuesday. What else? There's one more thing. Oh, yeah. My daughter is sick. So when I'm done here, I'm going to go home and I'm going to be doing office hours from my phone. So I might be a little late getting on and I might have to get off at some time because I might have to take her to the doctor. So I apologize in advance if I'm hard to reach for office hours today, but I'll do my best. Does anybody have any questions before we get started? All right. So, can someone please confirm that, hold on, I'm waiting on my screen to fix itself now. Oh, goodness. Come on. There we go. Can someone please confirm that you can hear me and see the slideshow?
Yes, we can hear you and see you.
Great, thank you. All right, let's get back to it. So to remind you again, obviously chapter 3 is all about biological molecules, which are broken down into 4 major categories. There's carbs, there's lipids, there's proteins, and there's nucleic acids. We've covered carbs. We started lipids on Wednesday. Lipids themselves are broken down into 2 categories. There's fats, which are triglycerides, and there's also steroids, and steroids are what we're about to talk about. Also, on Monday, we talked about how most of what we're going to talk about this chapter, these macromolecules, are monomers made up of repeating polymers. We talked about that on Monday. And then when we talked about carbohydrates, we talked about those monomers, which were monosaccharides, and the polymers were polysaccharides. And then when we got into the fats, I had to point out that technically these are not polymers made up of repeating monomers, right? They are made up of different parts, but they're not technically polymers made up of repeating monomers. So the last thing we talked about was lipids, and you do need to remember that those are made up of a glycerol and then those 3 fatty acids. And like I told you, or when I mentioned that, you don't necessarily need to remember that it's 3 fatty acids. Just remember that it's a glycerol and then fatty acids put together. And now we're going to talk about steroids. And for the steroids, I'm not going to physically, or I'm not going to verbally or put into words and ask you what steroids are made of, but maybe I'll show you a picture of one on the exam and you should recognize it by its shape. So let's get into it. Steroids, again, these are under the category of lipids, just like fats, but they're different. They're different than fats. They have a completely different structure and therefore, as you might imagine, a different function. Because again, remember those fats, those triglycerides, there's that one glycerol molecule, those 3 long fatty acids sticking off, right? That's completely different than these 1, 2, 3, 4 rings. Again, if you were to put it into words and describe the general structure of a steroid, it is a carbon skeleton with 4 fused rings. And again, I'm not going to ask you like that on the exam. I probably won't ask you anything about the way it looks. But if I do, I would give you a picture like this or some version of this, and you would have to recognize that as a steroid. So all basic steroids have this basic carbon skeleton. They're all different because they all have different functional groups. And you'll see some. I'll give you some examples, like different things sticking off the skeleton. But they all have the same skeleton. So are there any questions on this slide? All right. One of the examples that your book gives is cholesterol. Cholesterol is a key component of cell membranes. And most importantly, at least for the exam anyway, cholesterol is the base steroid. So that base steroid, that cholesterol, that's used to produce all the other steroids. So it's almost like the blueprint. It's almost like when you buy some kind of basic house, one of those modular homes, and then from there you can do these extra things that have all these extra add-ons. That's what the base steroid is. That's what cholesterol is. So here's a good example. Here's what cholesterol looks like, and then you definitely do not need to memorize that structure. But you can see from that can be modified to become this, which is testosterone, or modified to become this, which is estrogen. And you can see they're very similar. And again, I always say this, but I know you're not chemists and you're not biologists, so you don't know what all these things mean. But you can at least just look at it and say, oh yeah, that's an oxygen double bonded there, and that's an oxygen bonded to a hydrogen single bonded to that carbon, right? So you can just look at it and tell that it's different. And as long as you can tell that it's mostly the same but a little bit different, you can probably correctly, in this case, guess that it is slightly, that it's a very similar steroids, but slightly different function. But again, what you need to know is that cholesterol is the base steroid. That's the most important thing as far as the exam is concerned on this slide. So any questions about that? Okay. Now, a lot of times when people think of steroids outside of biology or chemistry or anything like that, outside of an academic setting, they probably think of synthetic anabolic steroids, which used to be associated with baseball a lot. So these synthetic anabolic steroids are variants of testosterone. They mimic some of the effects. They're used also to treat diseases such as cancer, AIDS, and other things. Like if you've ever had really bad poison ivy, for example, you might go to the doctor and they might give you a pack of steroids. So it's a really common thing to get prescribed steroids. But also people abuse it. They abuse it to build up muscle quickly. I'm sure you've already, you've heard of that before. As far as the exams are concerned, there's nothing on this slide that you necessarily need to be concerned about. We're going to talk about synthetic anabolic steroids, but I probably won't ask you any questions. There might be some questions on the study guide, but probably not on the exam. These synthetic steroids are mostly banned because they have health hazards, and in sports, they give an unfairness, right? This artificial advantage. That's why, going back, that's a picture of Mark McGuire. I don't know if you remember who he is, but he broke some records with home runs, but it turns out he was using steroids, so it wasn't fair. So here's some of the side effects of steroids. Again, I'm not going to ask you any of this on the exam. There's you can get some violent mood swings, which is known as roid rage. You can get depression. You can get liver damage, high cholesterol, which makes sense, right? Because again, cholesterol is the base steroid, and this anabolic steroid is just a version of that cholesterol. So anyway, shrunken testicles, reduced sex drive, and infertility. So That's just to say it because it's in the book. And again, this will not be on the exam. But are there any questions about this on the exam? I mean, any questions about this slide? So if you're interested in this, especially maybe if you're a student athlete, you could look into this, maybe write a little bit more about this on for your independent work. So good, we're jumping into proteins now. Proteins, those are polymers of amino acid monomers. So I'll just pause and let you soak that in because you definitely need to know that. Because remember, the whole thing about these large molecules is you need to know the monomers and you need to know the polymers. So the polymer for a protein is made up of amino acid monomers. And I'll give you the name of the polymer later. But for now, just know that the monomer is the amino acid. Your book points out, not that I'm going to ask you this on the exam, but your cells are over 50% protein if you were to take out the water, because we know that they're a large, like 70, 95% water, right? But if you were to remove all the water, your cells are mostly protein. And that in itself should tell you that proteins must be very important to your cells. Otherwise, you wouldn't have so many of them. They are needed in almost everything your cells do. And that's going to become very apparent a little bit in this chapter and a lot in chapters 5, 6, 7, a little bit in chapters 8, 9, and 10. So yeah, you'll see how important proteins are. They are also the most sophisticated molecule in your body. They're very important. We won't get away from proteins for a long time now that we've started. Are there any questions about this slide? Here's some examples of proteins, the major types of proteins, and I'm not going to ask you this on the exam either, but you have structural protein. So hair is a great example of that. Fingernails, that's a great example of structural protein. These horns on that sheep, those are structural proteins. Then there's storage proteins. So you think about, eggs are full of protein. Seeds also have a lot of protein, which also makes sense because you're storing proteins needed to grow, right? And if you're an egg, you're going to grow into some sort of chicken or reptile or amphibian or fish, right? So you're going to need proteins to grow. Same thing if you're a seed. If you're a seed, you're going to become a larger plant eventually. So that's why we have storage proteins. Contractile proteins, I think this is what a lot of people think about when they think about proteins. When you're not sitting in a biology class and you say protein, a lot of people think of meat, right? And basically meat is muscle. So yeah, contractile proteins. Transport proteins, these are really important, and we'll talk a little bit about these in chapter 5, but transport proteins basically let things in and out of your cells. And again, that's very important. We wouldn't be alive without that, even at the cellular level. And again, we'll talk more about those in chapter 5. And enzymes, in my opinion, these are the big ones. Enzymes are things that help chemical reactions go, which really doesn't give it enough credit. There's so many chemical reactions that happen in your body that you couldn't live without, that just wouldn't happen without the enzymes. And yes, they help, but they help in an essential way. Like they're so important that those chemical reactions just wouldn't happen. So without enzymes, we wouldn't be alive. So again, in my opinion, these two are the most important, transport and enzymes. And we're going to learn a lot about enzymes moving forward. Are there any questions about this slide? All right. Then let's zoom in and talk about proteins in general, because there's so many different types of proteins. But we're going to talk about just the generic protein right now. We're talking about the monomers, which are the amino acids. So when I say that the monomer is are amino acids, there are 20 different types of amino acids, and that's a number that you should know for the exam. And I'm not big on numbers, but you should know in every living thing, there are 20 types of amino acids. So every protein in every living thing on Earth is some chain of amino acids, some different variety, which is a lot, right? I mean, 20 isn't a lot, but think about all the different ways you can mix 20 different amino acids up. So know that number. And I put a little star here because there's actually a lot more amino acids than that, but there's only 20 of them that are common to all living things. So there are some amino acids that really aren't even involved in living systems. And if you're into that, if you want to look that up for independent work, feel free to look that up for independent work and write a paper. Anyway, again, there's 20 different amino acids, but all amino acids have this one basic structure, just like the steroids we talked about. Remember I showed you those 4 rings? They all have that same structure, but they're a little bit different depending on what's sticking off of them. And amino acids are kind of the same way. All amino acids have this basic structure. There's a center carbon, and then off of that, there's a carboxyl group, there's a hydrogen, and there's an amino group. And every single amino acid, all 20 of them, have the same exact structure. And then this part, they all have the 4th part of the carbon sticking off is what we call the side chain. And that's the only part that's different. They all have a side chain, but the thing is every different side chain is different. So in a sense, it's the side chain that defines the amino acid, because every amino acid, again, has the exact same amino group, hydrogen, carboxyl group, and carbon. Before I move forward, I'll let you know your study guide asks questions about the structure of an amino acid. I don't require you to know that it's made up of an amino group, a hydrogen, and a carboxyl group, all center, and a side chain, all bonded to a carbon. What you do need to know is it's the side chain that sort of defines the amino acid, because all the other parts are the same. The only variable part is the side chain. So are there any questions so far? Okay. So this is basically what I already told you when I had the picture up. The 3 basic common groups, the 3 groups that are common to every single amino acid. Again, the carboxyl group, amino group, hydrogen atom. And the only part that's variable is the side chain. So again, it's the side chain essentially that defines the amino acid, because everything else is the same. I've already talked about this, so we'll skip forward. And here's again another picture. It's the same thing, it just looks a little bit different. Again, though, carbon, carbon covalently bonded to an amino group, hydrogen bond, I can't talk today. A hydrogen and a carboxyl group, and again, that side chain. Again, each one has a unique side chain. And not only does that kind of define it, right? Not only does that make it one of the 20, its own unique amino acid, but that side chain also gives it its unique chemical properties. And we'll talk about that moving forward a little bit. And here's what I mean by that. So again, none of you are biologists, none of you are chemists, so I don't expect you to understand all the ins and outs of what this side chain means and what this side chain means. But you can, just from a layman's term, just look. This is kind of has a big triangle shape, right? And this kind of has a little rectangle shape. And even that in itself means something, right? Because these things, we look at them as, molecules, and we might not think of them as 3-dimensional things, but they are the 3-dimensional things that actually fit together with other 3-dimensional things. So you can tell just by looking that this is going to fit into something different than this. And then if we dig in a little bit for things that we do know, even that you guys know from chapter 2, I can tell you that this is hydrophobic. So it would be repelled from water. And a bunch of these lined up together would not let water through, for example. And this is hydrophilic. So that thing would be attracted to water. And also these 2 things would repel each other, right? Because that's hydrophilic and that's hydrophobic. And again, this is just 2 of the 20. So think about all the other hydrophobic ones. All the other hydrophobic ones would be attracted to this one and repelled from this one. So again, the point is, it's these side chains that make them unique, and it's also the side chains that give them their unique chemical properties. Because imagine a bunch of these things chained up together, a long line of these. Yes, it's a long line, but they don't just sit there in that long straight line. What they do is they fold up on each other. And when you try to fold up on each other, it's these side chains that kind of dictate how they fold up. Because again, if you try to bend this to where these 2 are together, they would repel each other, and you couldn't bend them to where they're together. So any questions about this slide? All right. So what happens is, again, we've been talking about amino acids, and what happens is these cells link them together, right? They put all these amino acids together, like I just said in that last slide, into one big long chain. And those are technically called peptide bonds. So yes, they're covalent bonds, but for this, they're so important that biologists gave them an extra name. Like, yeah, they're covalent bonds, but also under that, they're also peptide bonds. And if you have a chain of amino acids put together, that is called a polypeptide. So let me pause before I move forward. I don't require you to know what a peptide bond is. Like, there won't be any questions about what a peptide bond is, but your book included it, so did I. And also I included it because I hope it helps you remember that when you put a bunch of amino acids together, it's called a polypeptide, and that's where it comes from, peptide. Because in my mind, it would be easier to call it a polyamino acid, right? Because like with the monosaccharides, with the carbohydrates, the monosaccharide was the monomer, and the polysaccharide was called a polysaccharide. So that made so much more sense. So here, I think it would make it more sense if we called it a polyamino acid, but we don't. We call it a polypeptide instead. Also, and this is going to be a little bit more important later, but I'll go ahead and introduce it now. So again, sorry, let me back up. A polypeptide is just like a string of amino acids. Like, if these were all amino acids, I don't know if you guys can see me. If these are all just individual amino acids, and then I'm putting them together with peptide bonds. There we go. Peptide bond there, peptide bond there. And now they're no longer a string, they're no longer individual polypeptides. I've strung them together, right? So this is now a polypeptide. Here's where it gets a little bit more complicated. Let's see if I have anything where I can do this. Yeah, this will work. I've only got one person in here, but how would you describe this? What is this that I'm holding? She said a piece of paper. I agree. It is a piece of paper. Now, bear with me, guys. The suspense must be killing you guys at home because you can't see what I'm doing. Actually, you can't either. Good way to start off a Friday, right? So suspenseful. I'm charging extra for this. All right. What would you call this? A paper airplane, yes. But you could still argue that it's still a sheet of paper, right? It's just now that I folded it in this particular folding, it's now considered a paper airplane, right? That's the relationship between a polypeptide and an actual protein. A polypeptide is just that string of amino acids. It's not technically a protein until it's folded and gets whatever shape it's supposed to get. And that's also why I included this picture here. That's some sort of scarf or Afghan, whatever. It's something made out of yarn. So if you have a long thing of yarn, it's just yarn. But then once you twist it the right way and weave it the right way, eventually it will be considered a scarf. It's still a piece of yarn, but because it's been twisted to the shape it's supposed to be in, you would then consider it a scarf. And again, that is the relationship between a polypeptide and a protein. A polypeptide is nothing but a bunch of amino acids strung together, but it's not a protein until it folds and twists and does its thing and gets its shape. And it's never random, right? We talked about those side chains. Those side chains dictate how that thing folds in on itself and how it twists or spirals, whatever shape it takes. So, are there any questions about this slide? Okay. Actually, this slide should have been 2 slides ago, but like you said, when we put these amino acids together, that is technically called a peptide bond. Hey, just like everything else we talked about, when we put 2 monomers together, that's called a dehydration reaction. But anyway. So your book also points out that there's thousands of proteins in your body. And if you want to, for independent work, someone answer me that question. That would be really quick independent work. It'd be like 2 sentences. You asked us exactly how many proteins are in the human body. I looked it up, and according to this source, there are this many proteins. So there you go. There's a quick and easy independent work for you. Look that up for me. How many proteins are in the human body? Yes, there are thousands. So then you have to ask yourself, all right, if there's thousands of different proteins, but we only have 20 building blocks, how is that possible? And your book gives a good analogy here that points out that the English alphabet uses 26 letters, which is, close to 20. So it's similar, right? 26 letters from which to build words. and 20 letters, and when we're talking amino acids, in which to build polypeptides. So in the English language, at least back when I made this PowerPoint, I had to look this up. I don't know if this is still the case anymore, but there are 1,025,109 words, all from those 26 letters. So imagine too, when we're only talking about 20 amino acids, but these amino acids, there are all no rules as far as how they can be put together, right? With letters, you almost always have a Q and then a U, right? And there's the I before E, except after C, those kind of rules, right? There's rules for the English language. And for the English language, you only get words that are so long, but that's not the case with proteins. There are no rules as to which amino acids can be put next to each other, and they can be as long as they want or as short as they want. But this is just for your own knowledge. None of this will be on the exam. But are there any questions about this slide? All right, moving forward. If you download this PowerPoint, you can watch this video. This is a great example. This is what we call the primary structure of a protein. And when we say primary structure, that means we're just talking about the amino acid chain, right? It's not the shape at all. Primary structure is, okay, it's this amino acid attached to this amino acid attached to that one. Primary structure is all concerned about which amino acid is attached to which amino acid. Moving forward. Again, it's the amino acid sequence that determines the three-dimensional structure. So I'll use this quick example that's not in your book. You can see all these things. You're probably wondering what it is. You don't need to know what it is, but like it says GLY and LLE and VAL, those are just names of the amino acids, right? So those are all a bunch of different amino acids bonded to each other. And this one, the cysteine, just happens to have a sulfur that sticks out of it. And that sulfur also happens to be attracted to other sulfurs. So the fact that this polypeptide has a cysteine here and a cysteine here, that means they're attracted to each other. So what used to be a long straight line of amino acids folded in on itself because these two things were attracted to each other. And that's just an example. You don't need to memorize that. The point here is, again, it's the amino acid sequence. So the fact that there was a cysteine here and a cysteine here in this example, that determines the three-dimensional structure of the protein, right? So again, the fact that this one happened to be here and this one happened to be here made this thing fold up and bend back up on itself. And here's the important part. Here's what you really, really need to know from here and moving on out for the next few chapters. It's the three-dimensional structure of the protein that enables it to carry out its specific function. So the most important aspect of a protein is its shape. Shape is the most important thing for a protein. That's how it does its job. So a lot of times this semester, we've already talked about structure and function a lot, but this is the most important with proteins. Proteins do not work properly if they do not have the exact right shape. Because, and this part you don't necessarily need to know, but I just want you to know why bullet point 2 is so important. It's because of bullet point 3. Nearly all proteins work by recognizing and binding to some other molecule. So nearly all proteins work almost like a lock and a key, right? They work by touching something else or binding to something else. And if the shape isn't perfect, they can't do what they're supposed to do. And as we learned from slides back, proteins are very important, right? So if they're not doing their job, if they can't do their job, we just probably would not be alive or have something seriously wrong with us. So are there any questions about this slide? All right, we're making good time. Here we go again. Again, this is just the primary structure of a protein. To me, it would be better if it was a straight line because the fact that it's curved might make you think that's important, but it's not. It should be a straight line, but it's so long it just wouldn't fit on this slide if it was a straight line. So anyway, again, primary structure, we're just looking at, all right, that the lysosine is bonded to the valine, which is bonded to the phenylene, which is bonded to the glycine. All we're concerned about at first is just what is bonded to what. And then for some reason, your book chose this one to zoom in on, this amino acid, alanine. So again, you got the carbon, central carbon, bonded to the amino group, the hydrogen, the carboxyl group, and then, of course, the side chain. Here's another way you can look at a protein. So this is the same protein or excuse me, that's just the polypeptide. But once it folds up and goes into its correct shape, then it looks like this. And for you guys, that probably looks like abstract art. But for a biologist, we know what these ribbons mean and these straight lines mean. So it looks goofy, but it all means something to biologists. And then there's the space filling model. So that's more of like what it actually looks like if you were to look at the molecule. That's a three-dimensional representation of what it probably looks like, or what it does look like. Anyway, there's nothing here on the nothing on the slide that'll be on the exam. Are there any questions? All right. So we keep talking about how important the shape is and how it's the amino acids that dictate the shape, because again, those side groups interact with each other. They repel each other. They attract each other. So it's very important. It's very significant. So because of that, if you have a slight change in the sequence of the amino acids, that'll change the shape, which will then, like I've already said, change or affect the ability of the protein to function, right? And your book gives a great example, and this is not it. Before I give the example, let's just use this as an example. So you can see this protein it's a spiral, right? And it's a spiral because of the way these side chains are interacting with each other. This one has a hydrogen right there, right? This one has an oxygen right there. And those things are hydrogen bonded to each other. That's what those little dots mean. So obviously, if you change this amino acid or this amino acid, those things wouldn't be attracted to each other anymore. And therefore, that spiral would kind of unspiral, right? So you would no longer have the shape. Same here. We have all these hydrogen bonds holding these things together. And if you were to change one of those amino acids, then that thing would fall apart. So great example. But anyway, the book example is hemoglobin. Hemoglobin is the protein that's in your blood, your red blood cells. That's the stuff that actually carries the oxygen. So obviously, it's a very important protein. Without it, we would be dead. And if you were to substitute just one amino acid in that hemoglobin, you get something called sickle cell disease. And this is just an example, so you don't need to know anything about sickle cell disease. So again, here's the original amino acid sequence. If you look very closely, you can see it. Lysine to valine to phenylene to glycine to arginine. You can see it if you look in. And then when that long chain of amino acids folds up on itself, it looks like this. That's a normal hemoglobin protein. And then that goes inside of a red blood cell, and then the red blood cell looks normal. But then here, and this is a real life example, even though you don't need to memorize it, when you change this one amino acid, just one, because of that, that thing doesn't fold up on itself properly, and you get a slightly different sized or shaped sickle cell, hemoglobin protein. And because it's misshaped, your blood cell is misshaped. And please remember back to chapter 1 when we first started talking about structure and function. The example your book gave was blood cells. So think about that. The reason blood cells are big and flat and kind of donut shaped is because they need to let oxygen in and carbon dioxide out, right? That's their function. So when you get this weird shaped thing, its function is messed up. And that's why it's called sickle cell disease. It's not just like, oh, you've got funny shaped blood cells. No big deal. No, it's because you have funny shaped blood cells that you have this whole long list of conditions that we'll talk about later in the semester. And again, all this because you changed one amino acid. But are there any questions about this slide? All right. Again, like we already said, this is everything we've already said when we were talking about that last picture. One difference in the amino acid sequence is enough to cause it to fold into different shapes. And again, shape is the most important thing for a protein. So if you alter the shape, you alter the function. And a lot of times that causes diseases. And it's associated with a lot of diseases. And that includes some severe nervous disorders called prions. If you're taking notes, no need to write anything down about prions, because we will talk about those in a later chapter. They're going to come back. So for now, I'm just introducing you to prions. There won't be any test questions about them. But let's talk about them because your book does. Prion is basically a protein in your brain that is misshaped. The bad thing about it is it's also contagious, and that when that misshaped protein touches other proteins, it makes them misshaped, as you can see from these pictures. And what happens is you get these clusters of misshaped proteins, and it basically puts holes in your brain. And it makes your brain look a little bit like a sponge, which is where you get this name. bovine spongiform encephalopathy, which is mad cow disease. Kuru, that's where humans used to get this because they would eat other human brains. You can look that up for independent work if you want. It's actually quite interesting. It's like mad cow disease, but for humans, and the way it got spread was cannibalism. So interesting stuff. Then you've got stuff called fatal weight loss in deer, elk, and moose. So if you're a hunter, you might be familiar with this disease. And again, if you're interested in any of these, you can look it up for independent work. But none of this will be on this exam because we're going to come back to prions later in the semester. But are there any questions about this slide? All right. Again, I keep saying this. Hopefully nobody misses this question because I keep saying it. It's such an important concept. A protein's function is based on its shape. So its shape is very, very important. And also, its shape is sensitive to the environment. What I haven't told you yet is, we've kind of touched on it, but I haven't really talked a lot about it. But in chapter 2, when we talked about chemical bonds, I said it takes energy to keep these things together, right? When we take this atom and we put it next to this atom and we squish them together, it takes energy to keep them together, right? And what I didn't tell you, though, was it also takes energy to take them apart. So in some cases, the energy could just be from heat. So the hotter things are, the weaker chemical bonds become. So if you see this, again, these dotted bonds or these dots represent hydrogen bonds. And if you remember from chapter 2, and you do need to remember this for the exam, hydrogen bonds are the weakest of the bonds, right? So if you add a bunch of heat to this, eventually those hydrogen bonds are going to break, and that spiral, just imagine like taking scissors and cutting that, cutting that, cutting that, cutting that, right? That spiral will just unspiral, right? It would just be a long, flat thing of amino acids. So that's how temperature works. Also, think about this. Let's remind you what a hydrogen bond is because you need to know that for the exam anyway. So this is a good time to remind you. If we say there's a hydrogen bond, what we're saying here, there's an oxygen that's slightly attracted to this hydrogen, right? And that's oxygen slightly negative. That hydrogen is slightly positive. All right, so that's a good review, right? That's what a hydrogen bond is. Slightly negative oxygen attracted that slightly positive hydrogen. That's what a hydrogen bond is. Here's another good review for something you're going to need to know for the exam from chapter 2. What is pH? What does pH measure? Acidity, good. So if I say something is acidic, what does that mean? Not necessarily like pH. If I say something's acidic on the pH scale, that means it's 7 or below. But if we're talking about what kind of things are in the solution, does anybody remember what that means? If I say it's acidic, what kind of things are in the solution? I hate to use the word things, but how about ions? I just didn't want to give it away. But what kind of ions? There's a lot of these ions in the solution if it's acidic. Nobody remembers? You guys need to study. This will be on the exam. If it's acidic, that means there's a bunch of H plus ions. Likewise, if it's basic, that's so hard to do. If it's basic, that means there's a bunch of OH minus ions, right? So here we have this nice little protein that's all wound up and coiled up the way it's supposed to be. Now imagine if you put it into an environment that's too acidic. What's going to happen is that positive hydrogen ion, completely positive hydrogen ion, is going to be attracted to that oxygen, right? So that oxygen is going to want to be with that hydrogen, not this one, because this hydrogen is only partially positive. This one's fully positive. So because of that, all these hydrogen bonds are going to break because that oxygen is going to want to be with that hydrogen and not that one. Likewise, if you made it too basic, this partially positive hydrogen is going to be very attracted to this completely negative OH minus ion, right? So it's going to break its bond with that. So I'm hoping that makes sense. That's how changing the pH could disrupt the structure of the protein. Are there any questions about that on the exam, or any questions about this slide? And again, this is a good review for the exam. You need to know what a hydrogen bond is. You definitely need to know that when we say something's acidic, that means it has a bunch of H plus ions. and something is basic, it has a bunch of OH minus ions. So if there's no questions, we'll move forward. I'm really hoping to finish this chapter by today. Yes, good. So we're done talking about proteins, and then we'll talk about the final category, which is nucleic acids. So there's no questions about anything we've covered so far, right? All right. So before we jump into nucleic acids, I will say this. We're going to scratch the surface with nucleic acids. I'm going to give you the, you can consider this a preview, because in chapter 10, we're really going to get into nucleic acids, mostly DNA, but we'll also talk about different types of RNA and things like that. So this is just a preview. All right. So with carbohydrates and lipids and proteins, we talked about what do they do, right? Nucleic acids is the same way. What do? They store information and they provide instructions for building proteins. You could think of nucleic acids as literally a manual, like an instruction manual on how to build a protein. So every protein in your body, the information on how to build it, it's stored in the nucleic acids. There's two types of nucleic acids. Excuse me, you do need to know this for the exam. The two types of nucleic acids are DNA and RNA. Technically, DNA is called deoxyribonucleic acid. RNA is called ribonucleic acid. But for this exam, you just need to know DNA and RNA because, like I said, we'll talk about nucleic acids in detail later in the semester. So for now, you can just memorize DNA and RNA. Today, when you send me an email for attendance, include the word Wake Island. You look that place up too if you want. We'll talk about that place later. That's where the federal government eradicated a bunch of cats, but that's a whole nother story. We'll talk about that when we talk about invasive species. But yes, when you send in your email for attendance, include the word Wake Island for a little bit of extra credit. Anyway, let's move forward. Gene. What is a gene? I'm going to introduce you to this. I don't think I'm going to ask you what a gene is in this exam because we're going to talk about genetics later in the semester. So this is just an introduction. A gene is a unit of inheritance that is encoded in a specific stretch of DNA, and it programs the amino acid sequence of a polypeptide. That's a really long, fancy way of saying it's a set of instructions on how to build a protein. Or, of course, also unit of inheritance. So it's a set of instructions on how to build a protein that you got from your parents and that you will pass along to your offspring. That's all that means. And again, we're going to touch on this later in the semester. So right now I'm going to kind of fly through it because for now it's just an introduction. These instructions on how to build a protein, they are written in a chemical code. And that chemical code has to be translated. Well, first it has to be transcribed, and then it has to be translated. But again, we'll talk about that later. And basically what happens is you're going from this language of nucleic acids to a language of proteins. So we're going from DNA and RNA language to the language of this amino acid is attached to this amino acid that's attached to that amino acid. And again, I'm flying through this. And if you're taking notes, you can just read this, but don't study it. Because we will talk about this in detail later in the semester. So here's what I mean. There's a long stretch of DNA. Just imagine that going for, if that's to scale, that thing is going for, I don't know, thousands of feet. Anyway, we zoom in. Here's one gene. That gene is read. It's turned into RNA. And then that RNA message is read, and that's turned into a protein. And again, this is a preview. This will not be on the first exam. Here's what you do need to know. So as always, we talk about monomers and polymers, right? So what is the monomer of nucleic acids? It is the nucleotides. And in a way, it's a lot like amino acids in that we're also going to talk about what makes up the nucleotide. And remember, we didn't do that with monosaccharides. We just said a monosaccharide, oh, that's a monosaccharide. We didn't talk about how the monosaccharide itself was built, but we are talking about how nucleotides are built, just like we talked about how an amino acid is built. So remember, the amino acid had 4 parts. It had the carbon, well, I guess you could say 5. It had the carbon with the 4 things around it, right? So with this one, we're talking about 3 parts, and you need to know these parts. It's a 5-carbon sugar, a phosphate group, and a nitrogen-containing base. But to keep it simple, because this is only a preview, I bolded the parts you need to know. The three parts of a nucleotide are sugar, phosphate, base. I just need you to memorize that because later when we actually get into details with this, you'll at least have memorized sugar, phosphate, base. And then later we'll really talk about what the sugar means or what the phosphate means and the fact that the base is nitrogen containing and all that stuff. But for now, the monomer of nucleic acids is called nucleotides, and the nucleotides themselves are made up of sugar, phosphate, and base. So any questions about this? Oh, I don't think we're going to make it. We might. Here's a picture from your book. Again, sugar, phosphate, base. So this is one particular nucleotide. It's a T, and you'll learn what that means later. And here's the same nucleotide, just drawn a different way. So this is really important if you're a chemist and you want to know exactly how it's built. And this is probably more along what you need to know. Sugar, phosphate, base. So here's something else you need to know for the exam, for this exam. The same way there's 20 different amino acids, there's 4 different DNAs, right? 4 different DNA bases. And the same way it was the side group that defined the amino acid, it's the base that defines the DNA. So again, there's 4 different types of DNA. It's the A, G, T, and C. Yes, it's adenine, guanine, thymine, cytosine. For this chapter, for this exam, you don't need to know the full name. Just memorize the letters A, G, T, and C. So again, I'm comparing it to amino acids. There's 20 different amino acids. There's 4 different DNA nucleotides. For the amino acid, it's the side group that makes it different than the rest of the other amino acids. For the DNA, it's the base that makes it different from the other DNA nucleotides. Any questions about this slide? All right, so here's what they are. Here's what they look like. There's A, G, T, and C. You'll even when we talk about DNA later in the semester, you'll never have to memorize the shape. And again, eventually you'll need to know the whole name, but for now, just know the letters A, G, T, and C. As everything else we talked about, when you bring 2 molecules together, that's a dehydration reaction. So that puts this nucleotide next to this nucleotide. You use a dehydration reaction. They are then covalently bonded to themselves, and it is then called a polynucleotide. So there we go. Now we know that the monomer for nucleic acids is a nucleotide, and nice and easy, what happens when you put a bunch of nucleotides together? You get a polynucleotide. So the polymer for nucleic acids is polynucleotide. So these are really easy. Just like monosaccharides are easy, it's all in the name. The hardest one, in my opinion, is protein. Like I already said, it's covalent bonds that hold them together. Technically, it's the sugar attached to the phosphate. But for this chapter, you don't need to know that. Though yeah, I'll leave it at that. But we'll talk about that later in the semester. That forms something called a sugar-phosphate backbone, again, because you got sugar and then phosphate attached to a sugar, attached to a phosphate. That's why it's called a sugar phosphate backbone. And then the bases themselves hang off the backbones like appendages. I'll back it up, kind of see. I'll show you more pictures. And again, for now, this doesn't matter. This bullet point doesn't matter until later in the semester. DNA is double-stranded, meaning there's like a string of DNA here and a string of DNA here, right? And they're just next to each other. So when you see this picture, let me back up. This picture, yeah, that's one molecule of DNA, but what that is 2 different strands of DNA, polynucleotides next to each other, and then it just twists. The bases pair up. A goes to T, G goes to C. If you're taking notes, take notes that you don't need to know that for this exam. There will be questions on it probably on the study guide, but not on the exam because we will cover this later in the semester. There we go. That's what I was saying. You can see this is one strand. This is another strand. So DNA is double-stranded. You can see it's the hydrogen bonds between the bases that keep those strands together. That's going to be very important later in the semester, but not for now. So there's some similarities and some differences between DNA and RNA, and you need to know them. First of all, they're all polymers or nucleotides. So that's how they're the same. The nucleotides are the same in that they have the same 3 basic parts, sugar, a phosphate, and a base. But remember, even with DNA, same with RNA, it's the base that makes it unique, right? And it's the same thing when it comes to the differences between DNA and RNA. First of all, and again, I don't think I'll ask you this on this exam. This will be more later in the semester. Let's talk about the three differences between DNA and RNA. RNA has a different sugar. That's why it's called ribonucleic acid as opposed to deoxyribonucleic acid, because RNA has ribose, DNA has deoxyribose. Read it, brain dump it. We'll come back to that on chapter 10. Also, for the bases, again, DNA has A, C, G, and T. RNA doesn't have that. RNA doesn't have a T. It has a U instead. So it has A, G, C, and U. And then the big one, and this is one of the easier ones to remember, RNA is single-stranded, DNA is double-stranded. So any questions about that slide? We're running out of time. Just zooming in. We're just zooming in here at the nucleotide of an RNA. See, that's a U instead of a T, so that's how you know it's RNA. That's the easiest way you know it's RNA. And then, yeah, technically, this is a different sugar. I wouldn't expect you to know that by looking at it, but yes, it's a different sugar. Yes, all right. Every chapter has the process of science. I skip it every chapter because I'm never going to ask you any questions about it. I highly recommend reading it. It's good to get your brain thinking scientifically. After you read it, if you have any questions, let me know, and we can discuss it. Also, at the end of every chapter, there's something called the evolution connection. And again, I will skip it every chapter because I'll never ask you questions about it. But again, I highly recommend reading it. There will be questions about it on the study guide. So read it. If you have any questions, let me know. And we did it. Yes, 8:50. All right. So we are done with chapter 3. Sometime next week, we're going to take the exam. Make sure you keep an eye on your emails because I'm going to post something on Google Classroom where we can all talk, right? We'll figure out when we want to take the exam. So keep an eye out for that. Does anybody have any questions? All right. If there, wait, somebody have something?
Yeah, I actually think I may have misheard you with the word that we need to email you with our attendance. What did you actually say? Because I think that I may have completely misheard you.
Wake Island. I also just typed it.
Oh, okay, perfect. Then I got it right.
Yeah, I'm glad. You know, the other day it was monkey wrench, and I know it's hard to hear, especially with this mask on. Some people wrote monkey Rick, which I got a real kick out of because, you know, that's a completely made up thing. So anyway, yeah, the word is Wake Island. All right. If nobody else has any questions, like I said, I'm going to have to be on and off of office hours today because of my sick daughter, but I will do my best. So I will see you then if you need me.